So we will move on to the material at exam 3. Just to also let you know, there is no final exam here, okay? Exam 3 will be the last one. The material, we talk about genetics, biochemistry, and applied microbiology. And then we will be somehow next week or so, or we will announce a class project, which is a presentation. So you have a group together, 2 to 3 students, and make a presentation in April. And also April, we will organize 2 to 3 lab practice. Because you heard about all this microbiology, you never get any practice, that's not good. So we're gonna go to the lab, we're gonna practice the gram stem, streak plating, isolation, somehow let them make it a little bit fun, okay? Otherwise you're gonna listen this lecturing the whole day, it's not the way to know the material. Okay, we're gonna move on to the genetics part. Some of you are science major, you already learned, but others coming from the campus, you may not be very familiar with these, so we have to go over. When we talk about genetics, microbial genetics, we always go through the blue plan, is we call it a central dogma. So what is the central dogma we talk about? Is the DNA go to the RNA, and then RNA go to protein. And this is you learned in your like biochemistry class, microbiology class, biology class, or even nutrition class, or other science classes. It's all the same for bacteria because they are using bacteria, especially E. coli, as a research unit. So DNA go to RNA. Before that, they do this circle. So we call it DNA replication. And the DNA go to RNA. This is what we call transcription. And RNA go to protein. We call translation. And this is the blue plan. So we're going to go over these one by one, and also we're going to talk about the structure of the DNA. Okay, so that's the first one we call the central dogma. Now, you may be heard about somehow this RNA can go back to DNA. This is what we call reverse transcription. So that's also could happen. It's why call it a supplement to central dogma. Okay, so that's the central dogma. Now, we want to introduce you with DNA. We also want to give you some of the history about the science of DNA behind it. So let's look at this first slide first. So when people know about the DNA, it is around 100 years ago. The reason they started to do is the 1912 Spanish flu. Now, by the time they want to develop a vaccine, Spanish flu, it is very similar, the same level as coronavirus. The only thing is that it's killed more than coronavirus, and millions of people killed in the European countries. But by the time they want to develop a vaccine, so lots of people are working. And in 1929, a scientist called F. Griffiths, and he did a research trying to find transformation principle, or we say transformation concept, which means which material is transferring genetic information. So he did the study is on the mice. So what happened is there is Streptococcus pneumoniae. And Streptococcus pneumoniae, there is a two different type, and you can see here. One is a smooth one, another one is a rough one. So the smooth one is relatively a pathogen. So pathogenic. And the rough one, like that, it is a surrogate. So what this means, they do not have a toxication. It's non-pathogenic. And he used mice as a tool. So what happened is, I have a smooth Streptococcus pneumoniae. Okay, I injected a mice. Let's just say like this. The mice is dead. And we know that because it's caused pneumonia. And in the second experiment, he used a smooth one. Used a rough one. And he injected the mice. The mice is alive. And then what he did, he used the smooth one. Okay? However, he used a smooth one, but heat injured, which means heated. And then injected the mice, infected the mice. The mice is alive. Because the toxication material has been heat treated. We can say the bacteria is heat injured. Then the next one is very interesting. He had a heat injured smooth Streptococcus pneumoniae together with a rough one. And then inoculated the mice. And what he find is a dead mice. So what this tells you? This tells you there is something happened from the heat injured smooth cell to the rough cell is transformed there. So we know this is genetic information. There's something happening. But what it is, he doesn't know. Now, in the next 15 years, he continued to do the research, and he used the mice. So thousands of mice has been killed doing the research to find what's their genetic information, what's the material it is. However, he didn't finish the work. He was killed in the German bombing during the Second World War. However, other scientists have to pick it up. Then comes about here. This research is important. And we're not going to go very detailed, okay? We just briefly tell you. So, 1929, F. Griffiths finds that something is called genetic material there to do the transformation. He did lots of work in the mice that didn't find it, and he was died. Then, in 1944, there is 3 American scientists, Avery, McCarthy, McLean, at Rockefeller Medical Institution at New York City. They continue to using the biological methods, try to find the genetic information. And this time, they are not rely on animals. They did this in vitro, which means use tubes. Now what they find, They are using a combination of DNase, RNase, and protease in the tube research, and they find this genetic information is DNA. And they give the isolated DNA. So what they do, you can see the type R cells with type S cell DNA extract, they get DNA. But once they use DNase, they did not get DNA because DNase can hydrolyze DNA. If they use RNA, RNase, protease, the transformation still happens. So which means it is not a protein. The genetic information, the transformation principle is not protein, it's not RNA, it is DNA. And the report saying they got the isolated DNA. Okay, how purity it is, we don't know, but they'll get a breakthrough. They know the genetic information is DNA. Then, more and more research has to do. And this is done by two scientists in 1952, which is Hershey and Chase research. Okay, by the way, this Hershey is not a chocolate bar, not a candy bar. That's a scientist name. Alfred Hershey. And then Chase is not Chase Bank, it is Martha Chase. Two great scientists in 1952, what they do? They're using a different tool, not in the animals, not in the tubes, for bacteriophage. So it is very interesting what happened is, this is a bacteriophage, what it looks like. Very briefly. Okay, bacteriophage, the outside is capsid, which is a protein, the inside, it is DNA. So the idea is that this bacteriophage, they could inject a bacterial cell, and the bacterial cell could be bursted. And then we have to label the chemicals. So what is the specific chemical for DNA? It is phosphate. What is the specific unique chemical for protein? It is sulfite. So he labeled DNA, label phosphate 32, and the protein using S35. So he labeled them and he mixed them. Okay? Once you mixed them, what happened is he tried to separate them. And by the time there is no centrifuge. So what they have to do, they do in a kitchen blender. The blender actually will make juice these days. The principle is same as centrifuge, but they don't have to use a kitchen blender. And when they blend it, and they do a separation, what they find, and you can see clearly here, is in the bacterial cell, they find the protein. And in the bacteriophage, they find S35. And then here, what they find, the bacterial DNA is phosphate 32. They did not find the S35. So what this tells you? This tells you the genetic information is DNA. Because the phosphate 32 find in the bacteria DNA. And we just said the phage can integrate it. This part goes here. And this is labeled as phosphate 32. And I will show you why we do it. That's phosphate 32. And he did not find S35 in the bacteria cell. So this tells you the genetic information is DNA. So let's talk about the DNA. What does the DNA looks like? Okay. The DNA is a very boring chemical, I can tell you. So how are we going to say the boring chemical? So what is DNA? DNA, the structure is very simple. We have the phosphates. Sorry, we have the sugar. This is what we call 5-carbon ribose. And you can label it, okay? 1 prime, 2 prime, 3 prime, 4 prime. And then what happens is here, there is a base. And the 2 prime originally is OH, but then the O is gone. That's what the D stands for. Deoxynucleic acid. That's what the D for. That's O is gone. Okay, go here, you have H. Then you go here, CH2, O. How about here? It's a phosphate. So what you find in DNA? DNA composed by base, 5-carbon deoxyribose, and then what? A phosphate. So we say this is a very boring chemical. Compared to what? Compared to protein. Protein, you have amino acid sequence. Then you become a peptide. Then you have that's a primary structure. Then you have a secondary structure, like alpha helice, beta sheet. So you have a tertiary structure. Then you have a quaternary structure, which is a subunits. It's more complicated. DNA is very boring. Only have a base, deoxyribose, and a phosphate. Now, what are the base? ATGC. That's adenine, thymine, guanine, and cytosine. And how they connected with each other is A complementary with T and G complementary with C. And in the middle, what we call those chemical bonds, it's connected with hydrogen bond. So we're going to be doing a little bit of review. What are the 3 major chemical bonds we learned before in other classes? Let's say this is a water. Okay, this is another water. What are the bonds here? That's covalent bond. Covalent bond is very strong one. It's not easy to break down. You need energy to break down. The energy is very high. Somehow 150 kJ per mole. But how about the bonds connected here? That's hydrogen bonds. That's a weak bond. That's the reason the water can move in. You can break them. You only need 5 kJ per mole. Very simple to do it. What else? And so we learned the covalent bonds. We learned the hydrogen bonds. What else? Another bond you learned? Did you ever know this guy? Sodium chloride. How they connected? We call it a Lewis structure. They connected with positive and negatively charged, so we say it's ionic compound. Is that right? That's 1, that's 2, that's 3. We talk about chemical bond. And here it is a hydrogen bond. Because the hydrogen bond is not a strong bond, so they can be separated. But did you notice that GC has one more bond than AT? This is when we design the primer in the future during the polymerase chain reaction, like PCR reaction, we need to consider the content of G and C because there are 3 hydrogen bonds. Okay, that's very simple at DNA. So what's a DNA's full name? It's 2' deoxynucleic acids. So that's something we want to know. It's very important. And we have a picture here which tells you the development of that. So we have a gene base which is purine and pyrimidine. So A and adenine, guanine is purine, thymine and cytosine is pyrimidine, we call it. So A complementary with T, G complementary with C, they become the gene base with ribose deoxyribose become a nucleoside. The nucleoside with the phosphate there become a nucleotide. Then the nucleotide become long and long chain going through the polymerization process, become nucleic acids. And we will talk about the DNA, nucleic acids, how they synthesize. But this is just let you know how the DNA structure looks like, and briefly, very. This is we already mentioned, adenine and thymine paired with 2 hydrogen bonds, and the guanine and the cytosine with 3 hydrogen bonds. This is what the DNA runs. They are running the opposite direction. Can you see it? And they connected with what is phosphodiester bond. And what this means, they are running the opposite direction. Their means is the sugar direction is different. And you can see very easy. The sugar direction, one is up, the O is up, another one is O down. That's running the opposite direction. Okay? In my major class, I usually spending a time to draw the whole thing, but here we just be very briefly, the DNA is running opposite direction. From the picture you can see one is 5' to 3', another one is 3' to 5'. What this stands for is the sugar position. The O one is up, another one is down. This is another picture which has showed you how the DNA looks like. It's called double helix. Okay? And the backbone is deoxyribose phosphates. Strands hold together by hydrogen bonds between A and T and G and C, and we call this antiparallel. Okay, DNA replication. We want to mention some history to you. So DNA replication, this is a history spanning a lot of time. People wanted to find what's a DNA replication looks like. How they do it. There are some history we could introduce to you. So we talk about the 1929. F. Griffiths. We talked already about 1944. That's Avery, McCarthy, and McLeod find the genetic information is DNA, and we also talk about 1952, the Hershey research talk about using phage to verify that it is a DNA, is a genetic information. So time goes to 1953. There is 2 major scientists, Crick and Watson. That's Francisco Crick and James Watson. So Francisco Crick, by the time, is just got a PhD from Indiana University. And he has one hobby is watching birds. So he called himself is a birds watcher. So after he got a PhD, what do we do? Go to find a job. Now by the time, what is the most, we call it the most developed scientist area, the country in the world is in Europe. Certainly it's in Great Britain. So he went to Great Britain. At Cambridge University, at the Cavendish lab, find James Watson. And James Watson is a physician, he's a doctor. He served in the army, worked a little bit in the Royal Society. They have about 12-year-old difference, but they get along very well. And by the time, they call theoretical biochemists, which means they don't do research, they just go there, they talk and think, try to find what's the principle of the DNA replication, how they do it. Okay, so that's a very, now, how they do it? By the time, it's very lucky. They find a great scientist at the Cavendish lab, which is Rosalind Franklin. And Rosalind Franklin showing them using X-ray diffraction find the DNA structure possibly is double helix. Could be like that. We know now how they determined it. They spent time in the summer 1953, had a lunch with another great chemist, Graf. Graf is a chemist from Cambridge University. The Graf also did the research to find the structure of DNA. And by the time he told them, what he found is in a different biological body, they could have a different amount of A and T and different amount of G and C, but the ratio of A and T and G and C is always equal 1 and 1. Then, bingo, it's double helix. Now, why this is important, I want to tell you one thing. By the time, there is another scientist. He also finds. So, you know protein, have amino acids, have secondary structure. Secondary structure is alpha helix. You definitely have heard about that. What it looks like this. Who find it? Linus Pauling. He is getting very close to that. But why he did not cross the finish line? Because he doesn't know the Graf rules. This is a very infamous Graf rules. A and T the ratio is 1 and 1, G and T the ratio is 1 and 1. But Linus Pauling has lots of other contributions. For example, he found the electron activity for the covalent bonds. And he also had sp3 hybridization, which is in the chemistry. And he got a Nobel Prize twice. One for the sp3 hybridization. Another one is for Nobel Peaceful Prize, because he sent a letter to President Eisenhower by the time to restricted using nuclear weapons during the war, especially that's after the Second World War and after Korea War. So he had his honors there. But let's go back to, because Shapirov rules, they get determination, ANT and GNC. So they get a publication. There is a one-page publication at Nature. Only one page, talk about the DNA structure, double helix. Then in 1963, the Nobel Prize awarded to James Watson and Francisco Crick, and the boss of Rosalind Franklin, Maurice Wilkins. Now, Rosalind Franklin, unfortunately, did not got any honor by the time because she passed away. She had ovoid cancer. She passed away in 1958. Otherwise, the Nobel Prize will be awarded for her also. So this is a little bit history. Now, when Francisco Crick and James Watson published the paper, find the DNA structure, they do a little bit of celebration. Where they go? In England, they have a couple of beers, go to pub. And they go to a specific pub called the Eagle Pub. And they go there, they tell everybody, we have a breakthrough in the biochemistry genetics area. We find the DNA structure. But nobody knows what happened. So he paid everybody £2 to buy a couple of beers. And later on, the Eagle Pub become a tourist area. Right now you go to Cambridge, England, you can visit the store there. They have a DNA structure, James and Watson. Have a couple of beers there by the time. So that's very interesting in the history. But it is a big breakthrough. And that paper, only one page, lots of people say that's the single most beautiful paper in the 20th century, 21st century, because of the DNA double helix structure founded. Okay, so later on what they find, the DNA replication is semi-conservative. So what's a semi-conservative? Which means there is a replication fork. They break down based on the gene complementary policy. Then they go ahead to synthesize and match another bonds which is matched based on gene complementary policy. Now, who determined it? There is another important research what we talk about. Yes, that is around the 1950s. Matt Mendelson and Frank Starr research. So what they do is we have a double helix, okay, like this. And I label these is using nitrogen 14. Now, of course, it's coming from E. coli, okay? Now, what they do, they let the medium, the bacterial medium, is filled with nitrogen 15. What's the difference between 14 and 15? Is their mass weight. So this is what we call isotopes. Is that right? We talk about, so you have a chemical we call proton, we call neutron, and outside is a crowded area, we call it an electron cloud. And the proton is always positively charged, this is negatively charged, so a whole nucleus should be neutral, no charge. So isotope basically is neutrons is different. So he let the bacteria grow in the media filled up with nitrogen 15, now what happened? Okay, the double helix is separated. And based on gene complementary policy, this goes up, then this is an N15, and this is N15. Okay, go next one, what's going to happen? You will see somehow like this, somehow like that, but more and more, these showed up. These are nitrogen 15, these are 14. If we do a couple generation later on, majority of these will be nitrogen 15. And what he did, he used all these, go to a tube with Sicilian chloride and do the centrifuge. He find on the top is nitrogen 14. At the bottom, the huge is nitrogen 15. So what this tells us, semi-conservative replication. Is that right? Otherwise, it should be the average. That's a way they prove semi-conservative replication. Okay. Now, what we're going to talk about that, we want to go over what the DNA synthesis comes out. DNA synthesis. Here we're using bacteria as a module. And we need to know, because it's simple, it's purified, we can say. DNA in bacteria is circular. They have a original place for replication, have a replication fork going through semi-conservative replication, and when they find that the signal is ter, which is termination, is stopped. In fungi, it is different. The fungi, the DNA basically is linear. Okay, you have to know about that. Now, this is a story happened at the DNA replication fork, and we wanted to introduce to you briefly. In the genetic class, this could come forever. Could teach couple weeks, and each step, each enzyme, and we will be introduced to you pretty briefly. So what happens? So DNA has two bonds, double helix. One running from 5 prime, 3 prime to 5 prime. Another one is 5 prime to 3 prime. Okay, they all have a story. Using here, we call replication fork. When they started to have the replication, they need a DNA protein group, which encoding DNA A, DNA B, and DNA C. So what is the DNA? What is DNA protein? Initiation, which is send the cell a signal. We start to do the replication. What is the DNA B? Helicase. And what is the DNA C? It's a holder. The helicase holder. What does the helicase means? Double helix. Helicase, we want to break it down. So we break it down. Now, we know the physics. They are always highly twisted. When all of a sudden you break them down, what happened? They are very easy to twist it back. So we needed to let them keep them separate. Therefore, we need something to attach to it. These called SSB. Single strand binding protein. to let them separate. Okay, once they let them separate, they started to do the replication. Now, how do they do the replication? I believe you heard about it in your biology class. The top band is running from 3' to 5'. And DNA replication, you should remember, the direction is always from 5' to 3'. So, on the top, it's very easy they do it naturally. Okay, let's say ATGC. Okay, they can do it naturally. Let's say here is TACG, they do it naturally. Now, once they do the replication, they need an enzyme. What's the enzyme? What is the process called replication? Polymerization. So they need an enzyme called DNA polymerase. Basically, DNA polymerase 1 or polymerase 3 do the polymerization. Now, who find that one? Is Dr. Arthur Kornberg, a great American scientist, find it. And he won the Nobel Prize in 1970s. It's a DNA polymerase, do the polymerization. How about this band? So we call this leading band. How about this band over right here? We call it a lagging band. Why? You cannot do it naturally. You have to synthesize some very small sequence. Of course, all of these need RNA primer at the top. Because RNA primer, there's no restriction, so this is RNA primer. Now, what's the name of these small segments? You learned in your other classes called Okazaki segment. Dr. Okazaki is a great Japanese scientist. He had a very short life, only 44 years old. If he's still there, he's going to be a Nobel Prize in 1970s. People already say that. Unfortunately, he passed away. So that's called Okazaki segments. Okay, once you have these Okazaki segments, what we have to do, we need to cut them. These are in the primer, we don't need it. We're going to cut it. What's the tool for cut these? DNA polymers. One. This is 1 and 3, basically 3. And then we need to connect them. So how we connect it? We connected these. Need another one, we call it DNA, like this. Now here is a question for you. Why 5' to 3', the lagging band have to have Okazaki segments? Why they cannot naturally to do the replication? There is a picture here showed you very important. Right here you can see 5' to 3'. There is a free available hydroxy bonds. But you go the opposite direction, there is no free available hydroxy bonds. That's a major reason why we need Okazaki segments in the lagging band. Okay, there's other reason which is talk about the phosphates, the energy level. We skipped about that. In my major class I talk about that, but we skip talk about here. The structural reason. Okay, now, once we finished the DNA replication, there are two issues remaining. Number one, topological issue. What is a topological issue? When you have this is done, they are supercoiled. We call it positive supercoil. That's not good. Positive supercoiled cannot do anything. It's just a twisted way. The biological body doesn't easy to do the further work. And we need to let it become negative supercoiled. And very luckily, we had the enzyme called topoisomerase. Topoisomerase 1 can solve the problem. And topoisomerase in E. coli, it's very specific. We call it a DNA gyrase. So that's the first thing we solve the problem. The second issue, fidelity issue, which means error control. So how do you know after the replication the error can be controlled very well? We don't want any mistakes, because any mistakes happens. What happened is a mutation. We don't want a mutation for DNA, because a mutation could cause other things like cancer, for example. So fidelity issue. How to solve the problem? So we want to say, if we don't do anything, the error is very high. How high it is? 10 to the power of -3. Which means there is going to be one error happened every time, which is 1,000 base pair. That equals one bacterial cell become 2 cells. They could possibly have one mistake happen. So what they do?